So good morning, everybody. Thanks for joining us for today's CIC lecture. I'm very happy to welcome Pablo Rusjan, who's a relatively new hire here at the Douglas Research Center. Pablo did his PhD in Argentina, in Buenos Aires, at the University of Buenos Aires, and his undergraduate training is in physics, and his PhD is in computational physical chemistry. In 2003, he moved to Toronto, where he started a postdoc at the positron emission tomography center under the supervision of Kshitij Kapoor, and quickly became staff at the PET Center of CAMH, in charge of all quantitative aspects of clinical PET studies at the institution. He has more than 17 years of experience working in the field and has explored several aspects of brain PET, including first in vivo use of novel radioligands in humans, acquisition and reconstruction, analysis of imaging data, and the pharmacokinetics of radioligands and their application to PET in clinical studies to explore disease progression, treatment targets, and drug occupancy. So with that, thanks, Pablo, for agreeing to this talk, and I'm looking forward to hearing about the work.
Thank you. So I will speak about my expertise, which is kinetic modeling of radioligands for brain PET. PET means positron emission tomography, not a domestic animal. For the people that hear my talk 1 week ago, this talk has some slides in common, but it's a different one, a lot more boring than the previous one, and more technical. In the positron emission tomography, in the way that we use the positron emission tomography for quantification of brain proteins, when we have an interest in quantify a protein in the brain, we find a ligand for this protein, and a radiochemist try to radiolabel the ligand, replacing, for example, a carbon by a radioactive carbon. The radioisotopes that we usually use for radiolabeled molecules are carbon-11 and fluorine-18, which are in stable nuclei that decay mostly via beta plus decay. This means they convert a proton in a neutron, and during this conversion, a positron is emitted. And the PET scanner doesn't measure, even when PET means positron emission tomography, the PET scanner doesn't measure positrons. Actually, the PET scanner measures the gamma rays that are generated from the annihilation of the positron with an electron around in the area. So along 1 or 2 hours that we acquire the PET image, there are millions of these gamma rays detected in the PET scanner. And later, with some algorithm, we get an image that has in each element of volume a concentration or a number proportional to the activity in this area on the brain. When you have an X-ray, maybe you can look at an X-ray, or maybe an expert can look at an X-ray, and they can do a diagnostic that a bone is broken. In the quantitative PET that we do, actually, this is not possible. This is an example in which maybe you can observe something in a PET image. Without intention, in a study of neuroinflammation in elderly subjects that we were doing with Antonio Strafella, we acquired this image in which there is something clearly anomalous in the brain. Probably it's a tumor. Maybe some expert can make a diagnostic. Maybe the image can be good to quantify the size of the tumor. However, we were interested in a study translocator protein TSPO, which is a biomarker of microglial activation. And I was not able to determine whether in this tumor there was some neuroinflammatory process going on, because what is the high color in the red color in the image comes from the issue that there is more delivery, blood delivery and radiotracer delivery in the tumor, and not really from the fact that maybe there is a neuroinflammatory process going on, or I cannot recognize the neuroinflammatory process because the effect of the delivery is a lot higher or bigger. So to make quantitative brain PET, rather than look at a picture of the brain, we look at a movie of the radioactivity into the brain. So for example, we inject a radioligand in the veins, and after that we start to acquire the PET image, and the radioactivity in the brain evolves along the time. So in this image, we can delineate a region of interest, for example, the striatum, and we can study how the radioactivity or the activity change along the time. This is called a time activity curve, and I will use the notation Cp of t along the talk to describe this function. We can look at a different region of interest, and we can get a different time activity curve, for example, the one of cerebellar cortex. And this is a radioligand called PHNO that binds to dopamine receptors. And it is known that in the cerebellar cortex, there is very little dopamine receptor or not dopamine receptor at all. But in the striatum, there is a lot of receptors of dopamine. So there is some relationship with the uptake into of a radioligand into this area and the concentration of the target protein in the area. However, it's very tricky to try to quantify protein of interest only from the time activity curve. Here is an example. Carbon 11-B is the radioligand that binds to serotonin transporter. And these are the time activity curves of the cerebellar cortex before and after a selective blocker of serotonin transporter in a very high dose. So there is very little serotonin, very low concentration of serotonin transporter in the cerebellar cortex. And basically, these time activity curves are not showing a change of concentration of serotonin transporter in the cerebellar cortex. These time activity curves are very different because what is happening in the delivery of the radioligand to the brain. When we inject the radioligand intravenously, before going to all the organs in the body, it goes through the lungs. There is a lot of serotonin transporter in the lungs. So in the baseline condition, the radioligand gets delayed in the lungs before going to all the organs. When we block the serotonin transporter, we block the serotonin transporter in the brain, but also in the lungs. So the radioligand does not get delayed in the lungs and go quicker to the organs. So these time activity curves actually are very different, but are quantifying exactly the same amount of serotonin transporter, which is 0. So what happened in the arteries is important to understand the delivery of the radioligand to the brain. So in the kind of quantitative PET image that we do, we acquire blood and we study the time activity curve of the activity in the blood, the time activity curve of the plasma. We determine what is the time activity curve due to the unmetabolized ligand in plasma. And in addition, when we are able, we measure the free fraction of the radioligand. This is which fraction of the radioligand is not bound to a protein and is free. So there is more than one way to make the study of the blood. I will present here the one that I usually use, which is based on automatic blood sample system, which is this device. This is an automatic blood sample system. It has a pump that pumps blood from the arteries at a given rate. There is a gamma detector which measures the concentration of activity in the blood that goes through the detector. And there is the laptop here. So this is the diagram of how this works. Basically, the pump the blood, and the blood run on the detector, and the detector measure activity in blood. However, this way to measure activity in blood is full of problems. One of the problems is that the blood needs to travel a lot to arrive to the detector. The other is that the blood is prone to clog the tubings or the lines that for which it travels. So every once in a while, we need to rinse the line to avoid clogging, flushing a mixture of saline solution with heparin. Other big problem of using a gamma counter is related to delay and dispersion. Sorry, automatic blood sample system. Due that it takes time to the blood to travel from here to here, and in particular from here to here, there is a delay between what we measure in the automatic blood sample system and what we should measure in the brain, that is our area of interest. And in addition, there is a change in the shape, which is a phenomenon known as dispersion. So we ran the automatic blood sample system only for 22 minutes after injection. That is when the activity in blood changed very quickly. After that, the activity in blood started to change slower. So rather than pumping continuously blood, we take a few samples along the time. And if we merge the information of the automatic blood sample system with the manual samples, we are able to make a time activity curve of the blood. Later, if we are able to characterize the concentration of activity in the blood and the concentration of activity in plasma, and we take the ratio, we can divide the activity in blood divided the ratio of activity in blood to plasma to have a time activity curve of the activity in plasma. And finally, if we are able to know what fraction of activity in plasma is coming from the parent compound I use for a metabolite radioligand, we can multiply these two functions, and we can have what we call the input function, or the activity, the concentration of activity in plasma due to a metabolite compound. So I have go through too quick on these plots, because my plan now is to discuss these steps one by one. This way to estimate the input function is full of pitfalls, and I will, in the next slide, show how I deal with them. So this slide is the only one that is not for the general public. It's only for, in case that in the audience there is some experts in pets and they will get boring with the rest of the talk. But this is the way that I manage the delay and dispersion. This is the head curve. I hope you can see what I mean with this. The head curve is the truth events in the field of view, and it's a very good approach of a time activity curve of the full brain in the initial part of the scan after the injection, but it's almost free of noise in a resolution of a second by second. This cure here in black is the output of the automatic blood sample system, what we measure with the automatic blood sample system. So I used this model to correct by delay and dispersion. I say that the head cure can be fitted as a one tissue compartment model with the input, using as input the activity in blood corrected by dispersion. So this is what I measure with the ABSS, and this is how the cure will be when it's free of dispersion. And I model the dispersion with this equation. This is the equation for the one tissue compartment model, and using the one tissue compartment model and the deconvolve time activity cure of the blood using the Langmuir deconvolution, I can get simultaneously a value for the delay and for the dispersion. I do a couple of extra simplifications. When I have a radioligand as FIPA that enter into the brain very quickly and I can see the vascular contribution very clearly, I keep only the first term, but I set the constant K2 as 0, basically telling that in the first few minutes after injection, the radioligand is not returning into the blood. On the other case, when I have a radioligand like SL25, in which I cannot see the vascular contribution, but I can see that the head cure start to go down, I set the vascular contribution to 0, and I keep K2. So I will return now to the talk more easier for the regular public. Another problem that we have with ABSS is that the radioligands get sticks into the arterial line. So the ABSS measures some residual activity in addition to the one due to the circulation of the blood along the line. This problem is can be partially solved using different materials for the catheters. And it's worse when higher is the lipophilicity of a radioligand. For example, PBR, carbon 11 PBR and carbon 11 P2I are very well known to be very sticky. And the radioligand that I will speak a little today, SL25, actually sticks into the arterial lines. So this brings a problem of calibration because there is a residual contribution in what I can measure that is there all the time. So now I will show how I estimate the ratio blood to plasma for a few radioligands. As I said before, in addition to the automatic blood sample system, I take manual samples of blood. I count the activity in these manual samples of blood with an automatic gamma counter. And this will give me concentration of activity in blood. I centrifuge the blood and I get the plasma. I take a sample of plasma. I count the activity in plasma and I get this value here. So dividing the two values, I get the ratio blood to plasma. When I do that for several samples, I can have a pattern of how this ratio changes with the time. Using the definition of hematocrit, I can have an expression to how this ratio should look. And basically, it should be a number higher than 1 minus hematocrit, and it will be 1 minus hematocrit when there is nothing of activity in the red blood cells. So this is the ratio blood to plasma for SL25, and we can see that increase quicker at the beginning and later arrive to a plateau. And the initial value is very close to 1 minus hematocrit. The interpretation of this plot is not that the SL25 is entering slowly into the red blood cells. What happened here is that the SL25 has a very high protein binding, very low free fraction. So there is a protein in plasma that is very attractive for SL25. So the SL25 prefer to be in plasma than in red blood cells. When the radioligands start to get metabolites, are the metabolites what are entering into the red blood cells. The situation of SL25 is quite particular. Usually the kind of pattern that we see is one in which the ratio of blood to plasma decrease with the time. The samples that you are seeing here for curve are the average of 7 samples and of 7 subjects. And usually when you look subject by subject, you cannot see any clear pattern. The ratio blood plasma for the most of the radioligand is fit with a bi-exponential. And this presents a big problem because we don't know what happened before the first manual sample from the time of injection. So this extrapolation could be quite dangerous, in especial when the random noise in the samples make this extrapolation to diverge. I mean, this sample is very low and this is very high. Eventually, we will have something that goes to infinite. In addition, we know that at time 0, probably there is not activity in the red blood cells. So probably the real truth is that the ratio blood plasma starts in a value here and goes very quick up and down. But we cannot characterize that. And finally, how I study the radiometabolites. I take a sample of plasma and I inject this sample in an HPLC. And after the HPLC column, I have connected a detector of radiation. So the result of the HPLC in the detector of radiation will show some amount of radioactivity or index of radioactivity as a function of the retention time. The retention time will be proportional to lipophilicity and opposite or inverse to polarity of the compound that we are looking at. So here I am presented in the metabolization of curve analyzed with HPLC for 3 minutes and for 20 minutes. I don't remember whether I say carbon-11 curve is a radioligand that binds to fatty acid and mid hydrolase in a reversible way. It's a non-reversible radioligand, make a covalent union with the enzyme that try to measure. So what we can see is that at 3 minutes after injection, the HPLC show a very big peak that corresponds to the compound that we have injected, basically curve, and 2 small peaks that corresponds to the radiometabolites of curve. At 20 minutes after injection, there is very little curve and there is a lot of radiometabolites. Calculating the area under the peak respect of the area under the curve, we can generate a plot like this one, which shows the percent of unmetabolized curve in plasma, how much fraction of the radioactivity in plasma is coming from curve and how much is coming from the radiometabolites. For most of the radioligands, this ratio, this fraction is easy to fit with the Hill function. However, sometimes the samples has very low activity and the percent of parent compound in plasma is very difficult to determine. For example, the green line here shows the output of the HPLC for carbon-11 SL25, which is a radioligand that binds to monoamine oxidase B. And we can see that there is so much noise in the sample of 80 minutes that is very difficult to determine the area under the peak and the area under the cure. There is here a radiometabolite, but it's difficult to see. So what happened is that the sample presents a lot of noise and is very difficult to know whether the Hill function actually is telling us or predicting the right value at 80 minutes. We cannot generalize that always the same function will fit the percent of parenchympane plasma for all the subjects with the same radioligand. Recently, I was doing a study with a radioligand for histamine 3 receptors to study a drug called pitolisant. And I observed that for many subjects before and after pitolisant, the parenchympane plasma, the fraction of parenchympane plasma has the typical Hill function behavior. But for one subject, the same subject, in both in baseline and in postdoc condition, the pattern was different. So if I try to fit this cure with the Hill function, I will introduce error in the calculation of the input function. I am not sure what was the reason for this different pattern. Maybe it's related to some impurity that had the injection regarding radioactivity, some radioactive impurity in the injection, or maybe to something else. But for this subject, I have to use a different function to interpolate the data. So once that we do all this processing of the blood, we will be able to have a time activity cure of the radioactivity in blood here in red, and a time activity cure of the parenchympane plasma, or in other words, the radioactivity in plasma due to the metabolized radioligand. Usually, this plot is in logarithmic scale. Usually, at some point along the scan, both cures will be linear in a logarithmic scale, meaning that the clearance of the radioligand from plasma and from blood follows a first order kinetic clearance. Well, remember these cures because this will be they will be key for the modeling of the radioligand and for the and in order to get an index of a protein of interest using a kinetic model. So to make a model, let's think in all the contributions of activity to a time activity cure of an anatomical region in the PET image. So inside of a region of interest, we will have radioactivity coming from the blood and radioactivity coming from the brain tissue. And between the blood and the brain tissue, we will have the brain-blood barrier. So as we already saw, inside of the blood, we will have radioactivity coming from the unmetabolized radioligand in plasma, and we will have radioactivity coming from the radiometabolites. In this chart, I am showing different the activity coming from radiometabolites that can cross the brain-blood barrier and that cannot cross the brain-blood barrier. In the brain tissue, we will have a potentially a similar scenario. We will have activity coming from the radioligand-free tissue or activity coming from radiometabolites that can cross the brain-blood barrier or are trapped in the tissue. We will start analyzing a reversible radioligand. So in the case of an irreversible radioligand, we will separe what is the radioligand specifically bound to the protein that we are trying to quantify, this compartment, and the activity of the radioligand coming to radioligand bound to something else, which we call non-specific binding. So now we will assume that we have a nice reversible compartment. A nice reversible compartment, a nice reversible radioligand. A nice reversible radioligand will not have radiometabolites that can cross the brain-blood barrier. And we cross the brain-blood barrier only by passive diffusion. In addition, a nice reversible radioligand will not be metabolized inside of the tissue. So this simplifies our model a lot. So now we will say that we can describe, that we can use compartments, and we can describe the concentration in each compartment with a function. And each compartment will mean the state of binding of the radioligand. So we have a compartment for free radioligand. We have a compartment for specifically bound radioligand, and we have a compartment for radioligand non-specifically bound. A nice reversible radioligand, in addition, will be selective. This means that everything that is non-specific will have very low affinity, and it will be a very weak binding that will reach equilibrium almost instantaneously with the free radioligand. This means always a fraction of this compartment will be non-specifically bound. So now we will say something else. We will say that the radioligand can move from one compartment to the other following first order kinetics. And the first order kinetics will be characterized by rate content. So we can write several differential equations like that. We will say the concentration of radioligand in one compartment, for example, in the specific compartment, is what enter minus what exit. So due that this is first order kinetic, what enter is a rate constant K3 by the concentration in the compartment from where the radioligand comes from. And what exit is a rate constant K4 times the concentration of this compartment. Well, if we write several differential equations like that, it's actually quite easy, provided that you know the Laplace transform, to came with an expression like this one that I have here. And these equations say that the time activity curve can be expressed as a function of the activity in blood that we measured before in the previous slide, the activity due to a metabolized radioligand in plasma, and the rate constant K1 to Kn. So now, if we feed the time activity curve with the activity in blood and the input function, we are able to find the values of the rate constants. And from all these rate constants, we are interested in case 3. Why? Case 3 describes the transfer of radioligand from the free compartment to the specifically bound compartment. If we think in vitro reaction of ligand receptor, we using the law of mass action, we can express the rate of association as a rate constant of association ligand receptor with times the concentration of available protein of interest times the concentration of free ligand. And this factor here is the same thing that the rate constant case 3. In other words, if we use this model to fit the time activity curve and to get the rate constant case 3, we have an index of the concentration of available protein of interest. So all this model gives a relationship between the radioactivity that we measure with the radioligand and the concentration of the target protein. So this is how the model is simplified when we merge the non-specific binding with the free binding. And in some cases, these two compartments, and it's called two-tissue compartment model. And in some cases, we cannot distinguish the two compartments, and we have to use a one-tissue compartment model, which is simple, even simple. Measuring the rate constant case 3 usually is unreliable. So rather than the rate constant case 3, we have defined magnitude, which is the total distribution volume, which is an index more stable for available binding. So the total distribution volume is defined as the ratio concentration in tissue to concentration in plasma as a time of equilibrium. The total distribution volume in PET doesn't have units of volumes. The units of total distribution volumes are milliliter of plasma water divided centimeter cubic of PET image. The total distribution volume can be expressed as a combination of the rate constants, and in both cases will be proportional to case 3 and to the available concentration of protein that we want to quantify. So these are the time activity curves for the putamen and for the cerebral cortex for SL25. And the continuous line is the fitting with the two-tissue compartment model. So we can see that this simple model can predict the time activity curve of the radioligand SL251188. And not only that, the total distribution volume that we get from these time activity curves for different regions of interest correlate very well with the known concentrations of MAOB in those regions of interest in postmortem brain. So summarizing, the total distribution volume of the radioligand is an index of the concentration of MAOB in the region of interest. Not only that, if we do the experiment twice in around 7 subjects, we get almost exactly the same result. So to summarize the talk so far, we have done several assumptions. We have came with a very simple model with only 4 parameters. We have make terrible decisions with the radioligand that is sticking to the ABSS line and with the extrapolations in the ratio blood to plasma. And we have had huge difficulties to study the metabolizations at late time points with these very low concentration of activity. But however, we were able to get an index of a protein of interest that is reliable and reproducible. I think that this is quite remarkable that we can simplify a so complex interaction of a radioligand with all the molecules in the brain with a very simple model. I will show now how the model is for an irreversible radioligand, which will allow me to expand a little the conclusions. So when we have a nice irreversible radioligand, what we will ask is that the radioligand is metabolized inside of the brain tissue, and one that is metabolized is get trapped inside of the brain tissue, and the radioactivity cannot return into the blood. In this case, we will not have a reversible specific compartment. Again, we can use the same thing that before, the compartment, the rate constant, the differential equations, and we can associate the rate constant K3 to the concentration of the enzyme that we are trying to measure. This is how a two-tissue compartment model irreversible look like. In this case, the second compartment is irreversible. The radioligand can travel only in one direction. And these are the time activities of cure in black for the prefrontal cortex, in gray for the putamen. The one tissue compartment model does not fit the radioligand. For the putamen, the two tissue compartment model irreversible fit very well, but it's difficult to see. But for the prefrontal cortex, the reversible two tissue compartment fit the time activity cures better than the irreversible two tissue compartment. So even when we have an irreversible radioligand, the two tissue compartment reversible sometimes fits better. This time activity cure is for a single subject. This result can be generalized using some goodness of fitting criterion. And for 90 minutes data, the same problem happens for other regions of interest, like the temporal cortex and the insular cortex, when many subjects I take into account. So what is going on here? Well, we are violating some of the assumption of the model. We cannot use the two tissue compartment model to analyze this irreversible radioligand because we cannot find any parameter that is reliable as an index of a protein of interest. So I took here a decision, and it was a subjective decision. That is that the two tissue compartment model irreversible is the best model for this radioligand. And this is because it gave me a good, reliable, and reproducible quantification parameter. In addition, if I reduce the length of a scan to 60 minutes, I can calculate the same outcome parameter without an increase in the variability and without a bias. So, and when I have only 60 minutes of data, the two tissue compartment irreversible model is the best model to fit the time activity cures. So I determined that the optimal model for this radioligand is the two tissue compartment model with 60 minutes of data. I didn't feel comfortable with my decision before having done an experiment with a FAIN inhibitor. So in that way, I was able to see that the predictions of the model were good in the real life. So this is the time activity cure in baseline condition. This is the time activity cure after a high dose of FAIN inhibitor. And it's possible to see that in red and yellow, the two tissue compartment model irreversible and the one tissue compartment model reversible fit the time activity cure in blood condition well. And this is because In block conditions, we don't have this irreversible compartment, and we only have one reversible compartment. So to summarize this experiment and to summarize this talk, it's very common that the assumptions of the model are violated. So we need to take some decisions. More than one model can be good to modeling a radioligand and to get a parameter to quantify a protein. However, we have to understand how the assumptions could affect the parameter for quantification in different experiments, and we have to be careful how we make the interpretation of the results based on the parameter that we choose in each experiment to quantify the protein of interest. To finalize this talk, I want to say that I have automatized all the steps in the processing and in the quantification of the image in a set of software. So for a regular user to get an index of a protein of interest starting in a PET image can be reduced to to click with the mouse a few times in my softwares. Each software have a different purpose. This is to the linear region of interest, that one. This is to create the input function, and this is to feed the kinetic modeling. I would like to acknowledge many of my collaborators. The list is too long. I have chosen here the ones in which the collaborators with whom I have collaborate more in the more recent times. Well, thank you for stay here with my awful English. This is everything.
Thank you, Pablo, very much for that talk. We have time for a few questions. Are any questions from the audience?
I have a question.
Yeah.
If no one has a question, I have a question.
Okay.
So I would like Pablo, maybe you can tell us a bit what you think about the the quantification of the free fraction in plasma. I know we haven't done much of that before. I don't know. Can you share your thoughts on that?
Yes. If we have protein binding, if the radioligand is bind to protein, eventually will not be able to leave the capillaries and to enter into the tissue. So this could affect the outcome parameter strongly. So the best way is to correct the outcome parameter for the free fraction of the radioligand, the amount of radioligand that is not bound to protein. And the problem is that measuring the free fraction is very difficult and introduce a lot of noise in the measurement. I hope that here in Montreal, with all the equipment that we are buying, we will be able to determine the free fraction more reliably. However, this problem is a well-known problem in all the PET centers in the world. So there are, when it's possible, the best way, rather than to correct the free fraction in each scan, the best is to compare the free fraction between two groups or before and after a treatment. And if there is not a significant difference between groups, we can ignore it. I hope this answered the question.
Thanks, Pablo. Jamie, did you have a question? I saw you.
Yeah, I have a quick question just about sort of the practical aspect of doing PET quantification. So are there software packages out there that you would recommend for someone getting started with PET, maybe that would sort of have some built-in models that you could use to quantify some typical PET data, or is this something that you really do need to just like know somebody who does PET quantification?
There is a lot of softwares available around to make PET quantification. I recommend my software because they are free and they are well built. They are as black box as the other software, but I know the black box very well, so I can tell exactly what it's doing.
A question that was actually in this exact same line, that was actually the same question. So with your toolbox that you design, so, you know, what type of data do we need? Like, is it kind of just dynamic data because this is really what you're looking at? Or, so let's say that, you know, Jamie wants to start a PET study at McGill, and then, so what kind of data do we need there? Because we really need the software, right? It's, you know, it's like if the expert can analyze the PET and then that's it, right? Like easily, I would say. So could you talk a little bit more about your pipeline?
Yes. Well, the pipeline was designed for the images that we had at CAMH originally, but later I exported to other PET centers, so I was adding functions to make compatible with the data that they have in other PET centers. Originally, the pipeline was restricted to a very few functions, but this software, ROMI, is a software that includes a lot of things, not only to the linear region of interest. The software, and yes, it can work with the static PET images, with dynamic PET images. It can work with parametric maps. It has included inbuilt software for making partial Gaussian effect correction, to make motion correction. Even the software, at some point I did a work in TMS, and it has an algorithm to determine the best place to put the coil into the head to focus the TMS coil in one particular area in the brain. So the software is very general and very flexible. Eventually, I need to add a little of code to be able to load an image without previous preprocessing in a different software.
Wonderful. And then what about the scanner at the BIC, like, you know, like the HRT? Is there any specific thing that you need to tweak before we use it, or this is kind of all taken care before, so then we're good?
The software is specially done for an HRT because
okay, perfect, wonderful.
I was using when I did it. But of course, I have used the software with images from other institutions in Austria or in Germany where they didn't have an HRT, and it works too. The validation of the software, it was validated to get PET outcome parameters. Basically, the region of interest were not validated to measure volumes, but rather were validated to measure time activity curves. So it could be wrong to use this software to measure volumes of areas in the brain. If we change the PET scanner eventually, or if we change the algorithm of reconstruction, eventually it will be a good idea to make sure that still we are getting good time activity curves. It's highly likely that yes, but double check is always a good idea.
Malak, is there another question before I ask one last question about the software?
Not that I can see, unless someone else has one.
Okay, just jump on to one last. So, I mean, are you thinking about developing a launch null peptide pipeline? Sorry? Are you thinking about, I guess this is for cross-sectional data, right? Are you thinking about developing a longitudinal PET pipeline to see change, for example, in accumulation of amyloid or tau, or because this is way more tricky, right? So is it in your plan to kind of build that, or?
Well, there is a number of ways that you can do and longitudinally experiment. I recently was reading a paper where they did the longitudinal analysis based on a region of interest results. So basically, you can have two approaches. One is you try to do a parametric analysis. This is the voxel by voxel analysis where you look all the image as a whole. The other way could be you quantify the total distribution volumes of some region of interest, and you look only to the values of these regional parameters. So the software, if you're going to do the regional parameters approach, you don't need to have a graphical software. You can put the regional parameters in, for example, a neural network and try to get something. If you are going to do a parametric approach, probably, and this is quite interesting because it goes to a full field of the parametric maps, but probably the best way is to have a parametric image and later try to feed, for example, a convolved neural network to try to have some finding. But it's like independent of the software. It's like a step that you gave after dealing with the initial quantification of the radioligand. Whatever, this software, the ROMI can generate parametric maps that eventually you can use later in the convolved neural network to try to discover something.
Thank you.
But, the answer is so ambiguous because we have to define what is a parametric map. And this is the question. So sometimes an SUV ratio image is a parametric map. Sometimes a parametric map is an image in which each voxel contains a total distribution volume or a binding potential. And if you want to have an image in which each voxel has a total distribution volume or a binding potential, you have to do the kinetic modeling of the dynamical PET image first. So this is what the software can do. If you are going to use an SUV ratio image as a parametric map, this software can do it too. But later you will have to deal with the outcome of that.
All right, I think we're a bit over time, so I think it's a good time to end the talk. Thanks again, Pablo, for sharing your work with us, and looking forward to seeing how this all translates at the Douglas. Thanks.